Hello, everyone. So I am trying out a new system where I'm using my earbuds to record, and I hope that will reduce the background noise. So you guys should feel special. I bought these just for you. Wink, wink. Actually, they're just a rip-off brand. They're not the real earbuds, but that's okay. So first off, we're going to be talking about different ways to increase or decrease gene expression. And that means eventually, ultimately, increasing or decreasing the amount of a certain protein from a certain gene or a group of genes in the cell. And we were going to be talking about transcription factors. Now, we've introduced this word before, but we haven't talked about it too much. And so, let's see if I can move down here. Um, and I know this takes up part of the screen, but I want you guys to feel like you have a real teacher here instead of just being taught by a bodiless voice on the internet. Um, so transcription factors, they are proteins. So they're made up of amino acids. And there are different types of transcription factors. One type is a transcription regulator. We've also mentioned this term before, and a transcription regulator is also a protein. And we see here that most of the protein is just shown as kind of a blob, but we see one functional group from one amino acid projecting here, and this is asparagine, or rather the functional group of asparagine, and it is interacting, forming hydrogen bonds with two base pairs here. And so we don't see a lot of the DNA. The circle kind of represents the extent of the helix of the DNA, but we do see these two base pairs, and we see that this transcriptional regulator is forming hydrogen bonds with these base pairs. And so this is only one part of the interaction between this protein and the DNA. As it says here, there are often around 10 to 20 contacts or 10 to 20 different types of interactions, whether that be hydrogen bonding, ionic bonding, van der Waals effects, those types of things. Those all will make up those 10 to 20 contacts with different amino acids and different base pairs between the protein and the DNA. And in this way, the transcription factor is going to somehow affect whether or not that section of DNA is going to be expressed. And you can see, because of the chemical interactions here, how a transcription factor can be sequence specific. And it can be more or less sequence specific depending on, how many different contacts there are and how the contacts form. So we won't get too much into that. That will be something that you could talk more about in advanced molecular biology courses. But you should be able to look at this image and know approximately what's going on here. That the T and the A are from DNA, that this thing up here is a protein, that this is a functional group. We have hydrogen bonds forming between the DNA and the protein. All right. And so that's going to apply for a lot of the additional stuff that we talk about in today's lecture. So first, we're going to start off kind of slow with simple control of bacterial gene expression. And we're not talking about transcription right now. So transcription factors are something that we're going to talk about in a couple minutes. So put that aside for a moment. Here we've already transcribed the mRNA. So we have the mRNA, it's in the cell, and there are different 3D structures that some mRNA can form in bacteria. And as we mentioned yesterday, there are binding sites on the mRNA that the ribosome recognizes, and we actually have other binding sites that other proteins can recognize as well, and we'll be talking about that now. So here we see the ribosome binding site in yellow, so that's one that we talked about previously, and then that is followed by the AUG, which is the start site, and it codes for what amino acid? Methionine. So upstream of the AUG, the start site, we have the ribosome binding site, and that's where the ribosome is going to bind, and then it's going to begin translation at the start site. And we see here that there is this funny looking structure upstream of the ribosome binding site, and this is called a hairpin structure. So you might have heard this term before. A hairpin structure is any time that you get a structure where you kind of get like it's also called a stem and loop structure. So oftentimes you get complementary binding between nucleotides at one part, and then you get another part where there's no complementary binding. It just kind of like blebs out. So that's what this is here. Your hairpin or stem loop structure. It is, again, the thing that is sticking up. It looks like a lollipop kind of. And the region that has the complementary binding can change. So remember, these hydrogen bonds are weak, and these two strands could be pulled apart. And so you could actually switch between this structure and another version of the structure. So I guess that they don't actually have the other structure in the book. But I do have some pictures that I drew on the board last year, and I think that I drew them because the book didn't have the picture, or I didn't have it in my slides. And so this, where the base pairing occurs, can move back and forth between this place and the region that actually includes the ribosome binding site. So this is the version we just saw on the slide. We have the ribosome binding site here, and the start site here, and the hairpin upstream of them. Then there's another version of the base pair binding, or complementary binding, where the complementary binding occurs between those nucleotides that form the ribosome binding site and the nucleotides upstream. So then the region that was in the stem loop structure previously would be over here and over here. So even upstream of the stem loop right now. I should have drawn a little bit of a longer mRNA over here on this left side. And what is the point of this? Well, now a protein can come in and bind to this stem loop structure so that it stays bound like this and doesn't go back into this form of the stem loop, and it prevents the ribosome from binding to this mRNA. So this is a form of control where the bacteria has already made this mRNA, but something changes and it doesn't need this protein right now, or it perhaps needs more resources, so it can't make this protein right now. And so it prevents the ribosome from binding to this mRNA, but it doesn't degrade the mRNA. It's not wasting that effort or those resources. It can remove this protein with some modification, some other protein at some point, and then the mRNA can go back to this form of the stem loop structure, revealing the ribosome binding site, so the ribosome can come in and begin translation. Okay, so I'm going to end this little video here, and then we'll continue with our study of operons in the next video.